Hello there, my 109 lab students. This is Dr. Jenkins. Here we are already on lab number 3, which covers osmosis and diffusion. Now you might be saying to yourself, osmosis and diffusion, boo. I'm here to tell you it's not as bad as you think. So that's why I'm recording these little videos, and listening to them and paying attention, taking notes will be very worth your while, because osmosis and diffusion isn't as tough as it sometimes seems. So we're going to turn that frown upside down. All right, so in this lab, you're going to open up the lab sheet, which is what I have here, and it goes through what we would usually be doing in the lab, part 1, part 2. And then, as you're already familiar with, there is going to be an accompanying lab assignment. So some people like to open up the lab assignment while you go through this, because then you can just answer the questions as you go through the lab. All right, so part 1 is just some definitions. And I know that you've talked about this in a lecture with Dr. Roberts, but let's just make sure we go through it again. So here we have our cell membrane. So imagine this is the inside of a cell and this is the outside of a cell. It could be a muscle cell, a nerve cell, a liver cell, any old cell. And when you magnify this cell membrane, it actually looks like this. So when we magnify that cell membrane, what we see is what's called a phospholipid bilayer. So we have 2 layers of these phospholipid looking guys, where both the tails look in. So it's a phospholipid bilayer. The point is, when we have a cell, it's a living, functioning thing. And because it is a living, functioning thing, we oftentimes need to bring things into the cell. If this cell is going to do its job, it needs oxygen. It needs fuel like glucose. And then, on the other hand, let's see if I can change color. Ooh, so many pretty colors to choose from. On the other hand, we also need to be able to get rid of wastes, like CO2 that builds up, or urea, whatever the case may be. So we have to be able to bring things into the cell and bring things out of the cell. In order to bring things into the cell and out of the cell, we can utilize either active processes or we can utilize passive processes. Active processes, as the name suggests, requires fuel. And the fuel of choice is something called ATP. But we're not going to be talking about that so much. We're focusing on the passive processes, which is pretty cool. You know, little oxygen molecules, they can passively move into the cell from areas of high concentration to areas of low concentration. That's what diffusion is. But the beauty is that it's passive. It does not take any ATP or other energy. Good, I'm glad you're with me. You can see everything in bold are the things you should be reviewing. So the 2 passive processes that we're going to be talking about are osmosis and diffusion. Diffusion is when things move from high to low concentration passively. That's diffusion. That's the example I gave here. If we have more oxygen on this side of the cell, so here we have 1, 2, 3, 4, we have 5 oxygen molecules outside the cell, and inside the cell we only have 1. So how is oxygen going to move from high to low? And this happens passively, which is a great thing because our body can use this and rely on it without having to put out extra energy. It's fantastic. Osmosis is simply when we have water that moves from high to low concentration. So it's the same thing. It's just water. So I could draw the same old thing, but instead water. So let's say we have 1, 2, 3. Let's say we have 4 water molecules outside the cell, and we have 2 water molecules inside the cell. Is there a concentration gradient? Yes, there is. So water will move from high to low concentration. Now there's more to it. In order to have diffusion, the membrane has to be at least semi-permeable, which the phospholipid bilayer is. In other words, there's little tiny pores that exist between the phospholipids. But basically, just stay with the basic definitions, osmosis and diffusion. And check this out, my friends. All, and I mean all, of our body organs rely on this. So we're not just going through osmosis and diffusion because we want to make our students' lives miserable. So let's have our students learn as much as they can about osmosis and diffusion. We're going to throw in as many technical terms as we can. No, we talk about osmosis and diffusion because our body uses it all the time. Right? I've given you some examples. How do we exchange gases in the lungs? Diffusion. How do we filter blood in the kidneys? Diffusion. And those are just 2 examples. So this process is happening all the time. We would not be able to survive if our cells weren't able to diffuse things into and out of the cell. Isn't that exciting? I think so. All right. Now, the first part of this lab, after you go through the definitions, would be to view these videos on something called Brownie in Motion. So as you go through the lab, you're going to click on the videos, you're going to watch Brownie in Motion. I've defined it here for you, so I'm not going to read off the page. Pretty easy concept, just be able to identify, just be able to define it, and then also watch the video so you see what it looks like. Now we get to the real sciencey part. Now that we understand what diffusion is, movement of particles from high to low concentration, and this process is passive, does not require energy, extra energy like ATP. Now that we understand what diffusion is, in the lab we can see what are some things that influence diffusion. So in this lab, we're going to look at how temperature influences diffusion, how molecular weight influences diffusion, and how a concentration gradient influences diffusion. We've got videos for you to watch. So the first one is looking at temperature, and I'm going to ask you to come up with your hypothesis as you go through the lab. Do you think that diffusion occurs more, or do you think it's easier to diffuse at a warmer or higher temperature? Well, watch the video to find out. Then we're going to be looking at molecular weight. So we have 2 different dyes in the video. One is potassium permanganate, it's kind of purple or red. And the other is methylene blue, which is blue. So the question to ask yourself is, do you think that a heavier or lighter weight particle will diffuse more? Now I want to make a point. This is molecular weight, which is not necessarily the same thing as size. So some people make the mistake of thinking, well, if it's lighter in weight, it must be smaller. But that isn't always true. Think about a balloon. A balloon can be lightweight, but it can be pretty big in size. So we're looking here at weight. So you'll see in the video, we have a blue and a purple or reddish color particle, and they have different molecular weights. One is heavier and one is lighter. When you watch the video, you're going to be able to see which one will diffuse more. And this matters, our body has understood what influences diffusion, and that's why your cells and your organs are made up the way that they are. Or if we are trying to do something in the body and we know that temperature and molecule size can influence diffusion, we can use that to our advantage. Think about prescription drugs. If we want to get something from a pill you take into your cell quicker, then we can make sure that that molecule we have in your pill is the right, for example, weight. So we can use this. And then we're looking at the effect of concentration gradient. Now this one is a little bit trickier, and I want to outline it for you. And I have some notes. Let me just look to my side. Yes, even I need notes sometimes. Well, just because I have so many classes, I want to make sure I get it right. In this experiment, you're going to set up, well, in the video, pretend, we're going to set up 3 different beakers, and these beakers have different solutions in them. So beaker A has all water. Beaker B, that fluid, is a 15% sucrose or sugar solution, and beaker C is a 40% sucrose solution. So we have different solutions in these beakers, and into them we're putting, you'll see it in the video, it's a dialysis tubing bag. It's basically a kind of rubbery little tube that we tie either end, and we have a fluid on the inside. Well, these tubing bags all have the same concentration, right? So all of these bags have a 15% sucrose solution. Now, before you fall asleep, wait, pick your head up, don't fall asleep yet. Let's draw this out. And this is what I was saying at the beginning, when sometimes it can seem really confusing. And in a lab experiment like this, if we don't break it down, it can be confusing. But this is why you have me. I just took a sip of water because I mean business. So let's draw this out. Anytime, even me, right, teaching you, anytime I'm trying to think about diffusion, I like to draw it out. And I'm going to show you what I mean. So I'm going to have beaker A, beaker B, and beaker C. Now, as I said before, each beaker has a different solution in it. And then into that, we're going to put this dialysis tubing bag. Okay. Now, what we're going to be trying to figure out is, how is the water going to diffuse? We care about the sucrose, yes, but we're going to be looking more at the water because there's more water. So because there's so much more water in the beaker, and because there's more water than there is sugar, that's what we're going to look at to see what will diffuse. I think you're going to understand as we lay it out. So how I try and make sense of this is I'm going to look at the solution inside the beaker and draw it out. So what did we say? We said that beaker 1 contains 100% water. So now I know what this fluid is in beaker A. Beaker B contains 15% sucrose. And if this solution is 15% sucrose, I can figure out how much water is in that solution. If it's 15% sucrose and the rest is water, how much water is there? 85% water. In beaker C, it contains 40% sucrose. If the remaining is water, that means 60% of it is water. So now I've specifically laid out the concentration of water and sugar in the beaker solution, which is what's in blue. Let's figure out what's in red. And we already said this. The dialysis tubing bags all have the same thing. So we know that inside 15% sucrose, and if the rest is water, 85% water. So it's the same for all three. 15% sucrose, 85% water. 15% sucrose, 85% water. So now when you've laid it out like this, it's so much easier to see what's going to diffuse. In any situation, if it's a cell, I compare the inside of the cell to the outside of the cell. In this lab experiment, I'm comparing the inside of the bag to the outside of the bag. And almost always you're going to be looking at the water. Because there's more of it, that is what is going to really diffuse. So let's look at beaker A. If I ask myself what's going to happen with water, I'm just going to compare the water concentrations inside the bag to the water concentration outside the bag. If we look at beaker A, we have 100% water outside and 85% water inside the bag. How's water going to diffuse? Well, we know that diffusion happens from high to low. So water is going to diffuse from higher concentration to lower concentration. Cool, right? We just look at the water, compare the inside to the outside. Look at beaker number 2. 85% here, 85% there. What do you think is going to happen? Beaker 3, 60% water outside, but 85% water inside. I'm going to let you figure out how water will diffuse, knowing that water will diffuse from higher to lower concentration. And you can watch the video, which I thought there was a video somewhere. Yeah, there's a video right here. Video link. In lab, we would actually weigh the bag initially and then after it's submerged in the water. And let's look at beaker number 1. It says bag number 1, but really it's beaker number 1. In beaker A, water is going to diffuse from the outside to the inside. So what's going to happen to the weight of that bag? If water is diffusing into it, will that bag gain weight or lose weight? You can see the answer started here, it ended here. So you do the math, and you're going to calculate the difference between the two, and then whether there was a gain in weight, a loss in weight, or it stayed the same. Pretty simple. So all we're saying here is based on the concentration gradient, how much water is on one side versus the other, that determines how diffusion happens, because diffusion always happens from higher to lower concentration. Listen, if you take the time to watch this video, it's going to help make sense to you, I promise you. And as always, email me, come to my virtual office hours to ask questions. All right, now, the last part of this lab deals with something called tonicity. And this can be hard. And this can be hard even for students who are studying science all the time. This can be a tough concept. But I am here to help make your life easier. If you'll just give me a chance. So I'm going to get a sip of water here because I'm talking a lot, I'm getting into it. Oh yeah, I'm ready. Okay, there's this characteristic called tonicity. And I'll let you read this, but basically tonicity means how a cell may change shape. How a cell may change shape. If I have a red blood cell and water comes diffusing in like crazy, red blood cells are pliable, they can expand and can recoil. So in any instance where I have a lot of water all of a sudden diffusing into a red blood cell, wouldn't it make sense that that red blood cell may get bigger? Yeah. So based on what's diffusing into or out of a cell, that cell may change shape. And that is a characteristic called tonicity. This is different from this experiment. In the previous experiment, these tubing bags, they really didn't change shape that much. I mean, a little bit, but not to the naked eye. When we're talking about tonicity, we're really referring to cells in the body. And the best example of this would be our red blood cells. Because water frequently diffuses into and out of our red blood cells. But if we have a lot of water diffusing in at once, or if we have a lot of water diffusing out of a red blood cell at once, then that red blood cell itself may change shape. And the ability or the characteristic of how a cell may change shape is called tonicity. And we have 3 scenarios. It's really not rocket science. We just apply these words that make it seem hard. Anytime you have a red blood cell, this is a red blood cell, right? Anytime you have a red blood cell and it gets bigger, anytime a red blood cell gets bigger, the solution, the total solution, so in this example, this is the total solution, which really is akin to your bloodstream. Anytime a red blood cell gets bigger, the solution that it's in is said to be hypotonic. Anytime a red blood cell shrinks or gets smaller, that solution is said to be hypertonic. And then anytime a cell stays the same shape, it's called an isotonic solution. Iso means the same. It's a little confusing in the naming because, you know, the prefix hyper means more. So you might think a hypertonic solution is when a cell gets bigger, but it's actually the opposite. And I don't want to get into the specifics of why that is, but don't get caught up in that. Because in this instance, a hypertonic solution means that cells shrink. Hypotonic solution means that cells get bigger. The way that I remember this is kind of silly probably, but it works. I think about if someone is hyper, they may go see a shrink. A psychiatrist. If you can find these silly ways to remember things, it's more likely to stick with you. So I've always remembered this, even to this day. Hypertonic solution, hyper shrinks. That's really the basis. It's just understanding these definitions. In the lab, you're going to watch the video because you're going to actually see this happening. And it's pretty cool. So you're going to set up 3 solutions. So just like it shows here in the video, they set up these solutions. Now, what did I say before? What I said before was, anytime I'm trying to figure out how something's going to diffuse, I draw it out. And I'm going to do the same thing with you right now. And you can see up here, this lays out what we would be doing if we were in lab in person. And really what they do in the video, even though in the video, they don't explain it the best, but it's good because you can actually see the cells changing shape, which is cool. So here's beaker number 1, beaker number 2, beaker number 3. And we have a solution just like we did before. This is like my water symbol for a solution. Beaker 1 contains a 3% sodium chloride or salt solution. And because we understood this before, the rest is water, so 97% water. Beaker 2 contains 0.9% sodium chloride, which means that 99.1% of the rest is water. That may seem weird to you, like why did they pick 0.9%? Why did they pick 3%? Well, we can attach real solutions to this. A 0.9% is the same thing as saline. So the saline solution, someone puts their contacts in to keep them moist. Or if you get an IV bag of saline is this. A little bit salty. 3% is close, seawater, depending on where you are in the world. Seawater is usually, I don't know, 3 to 3.5% sodium chloride. You know, if you go to the beach and it's salt water, that's about 3 to 3.5% salt. So that gives you an idea of what these solutions are like. And then beaker 3 is 100% water. And now into each, they're going to drop blood. So imagine they drop blood, and that blood contains all these red blood cells. So just like before, we're going to compare the water inside the cell to outside the cell. How about that? Now, what do we know about blood? Well, blood is in a solution that we know to be constant. And the solution of blood is 0.9% sodium chloride, which means the remaining is water. That's pretty cool, right? So your red blood cell, the fluid that your red blood cells sit in, is a little bit salty. And the rest, because our blood is mostly water, but there's some salt in there. So once I'm done laying this out, now we just look, and we look to see the water concentrations. Let's look in beaker number 1. We compare outside the cell is 97% water. Inside the cell is 99% water. How is water going to diffuse? Well, water will diffuse from high to low. So in this case, even though it's not a big difference, it's still a difference. And folks, even if there's a small difference, there will be some diffusion. So water will diffuse from inside of the cell to outside of the cell. I'm going to let you write in what happens. So if a lot of water leaves that cell, what is going to happen to the shape of the cell? If a lot of water leaves it, will that cell become smaller or bigger? And then based on that, you can label whether this solution is hypertonic or hypotonic. You can do the same thing for beaker 3. I'm going to jump over to beaker 3. You compare the water outside the cell to inside the cell. Water will diffuse from high to low. But I'm going to look at number 2. In number 2, we have 99.1% water outside of the cell, and we have 99.1% water inside the cell. Guess what? They are the same, which means this is an isotonic solution because the red blood cells stay the same size. You're going to see a question in your lab assignment about why we want an IV bag. So if someone is dehydrated and we give them an IV bag, we absolutely want that IV bag to be saline. Because if it's saline, which is 0.9% sodium chloride, it is isotonic to your blood. So your red blood cells won't take in fluid and burst, and they won't get smaller. The other scenarios, in most cases, are bad. We don't want our cells to take on a lot of water and burst. We don't want a lot of our cells to get smaller because then they can't do their job properly. So this is why if you're stuck at sea, you don't drink the salt water as it is because your cells will, you'll figure it out, they'll either burst or get smaller. This is why we wouldn't want to give 100% water in the IV bag because it'll disrupt your red blood cells shape and they cannot do their job and you might die. All right, really good stuff this week, folks. I know if you look at this now, it looks complicated. Please, please watch the video again and again. Ask me if you have questions. I'm here if you're needed.